This is Miss D.M. Arvekar, working as Assistant Professor in the Department of Zoology, Rajarshi Chhatrapati Shahu College, Kolhapur. And in this small video, we are going to discuss about the enzymes. Have you imagined if you keep one piece of a food in the surrounding environment, what will happen to that food? Yes, obviously you are correct. As it will act upon by the external environmental factors, it will get degraded out. Similar type of a mechanism takes place in our body, but it will require a small duration of a time as compared to the first reaction as I told you. So what are the main reason behind it? So that whatever food we take inside our body via the digestive system, it will acted upon and it will be digested out into the simple form, and that simple form will get absorbed in our body and then it is circulated throughout the corner of the body by the blood. So we are going to discuss about these certain magical molecules which are present in our body or in the bacterial cell or in the plant cell, so that they are acting similar to the catalyst which we were using during the chemistry reactions or the chemical reactions. So in the chemistry, we have already studied that suppose the A and the B, these are the reactant, and they will react with the certain reaction mechanism, then it will be obviously converted into the product, that is C and C plus D. But if I am using certain catalyst over here, so that that catalyst will increase or speed up this rate of reaction, and so if suppose by half hour I am getting this reaction, but with the help of such a catalyst, I will get this reaction within the time or within the less time or within the 10 minutes, whatever that may be, depending upon the catalyst. But surely we are using this catalyst with this reactant, but it will never happen that it will react with this reactant. So obviously these are the very special molecules which are used in order to speed up the rate of chemical reactions. But such a type of a chemicals or the magical chemicals which will not take part in the chemical reaction, but they will surely increase the rate of a reaction. And that particular reaction or the molecules which are increasing the rate of a reaction, that are called as a catalyst. But similar type of a catalyst which are present in the living cell, which will catalyze or speed up the rate of a reactions which are happening in the living cell, or the biological reactions which are happening in the cell, that are catalyzed by the catalyst, and that catalyst are called as a enzymes. For example, if we are taking the food into our oral cavity, then it will be acted upon by the saliva. And that saliva consists of the salivary amylase enzyme. So amylase enzyme is a very specific, it will act on the starch if it is present in the given food material which is present in the oral cavity right now. And during the mastication process or during chewing process, it will mix with the saliva which consists of this salivary amylase. Then that salivary amylase will act on the starch and it will degrade that starch partially in our oral cavity. So number of enzymes are present in our digestive system, so that whatever food we will take inside, it will get simplified or it will be converted into the simple molecules, and the body will get such a type of a simple molecules, so that it will get the energy. In in today's world, the number or the thousands of enzymes are there which are used in every type of industry. In the detergent industry, it is a very famous because whatever the scars or whatever the dirt is present on the cloth, that can be removed with the help of a detergent. But why it is possible? Because detergent consists of the different types of enzymes which will act on such a type of a dirty material. It will act on the lipid, that is a lipase enzyme. It will act on the proteins which is present in the dirt form, that is protease enzyme. It will also consist of the amylase, and the such a type of a amylase enzyme or such a type of a enzymes are secreted with the help of a certain bacteria, and it is present or produced in the bulkier form, and it is used at various level of the demand. Similarly, the enzymes are also used for the preservation or maintaining the freshness of the meat For example, the leaves of the papaya tree is used to maintain the texture of a meat at the international level in certain countries. So, number of enzymes are used in our day-to-day life, and it is necessary to understand how that enzymes work, or what are the different types of enzymes are there, and how the reaction will get catalyzed without changing the chemical reaction. Now, we are not going to discuss about in detail the discovery of enzymes, but these are the certain contributors who have discovered about the enzymes or enzyme mechanism. The first one is Anselm Payen, who discovered the enzyme diastase, or also called as a zymase. And he discovered that in the cell-free fermentation process, and for that he got the Nobel Prize. Then the microbiologist Louis Pasteur, who told that yeast cell is responsible for the alcohol fermentation. Then the scientist William Cune first coined the term enzymes. Then the Edward Buckner, he told by his experiment, very famous experiment by using the yeast extract, he told that the fermentation which is not caused by the living yeast cell but the yeast extract. So he told that yeast cell is not responsible for the fermentation, but yeast cell consists of the enzyme that is important for the fermentation process. Then B. Sunner isolated the enzyme urease from the jacobin in the pure crystalline forms. Nowadays, you will get the different types of enzymes which are used at the industrial level or the day-to-day life or at the domestic level. Now, if we consider the chemical nature of enzymes, then all enzymes are proteins, but there are certain exceptions. That is, there are certain small fragments of RNA which are also catalyzing certain reactions to speed up the reaction. So, not all the enzymes are protein, but almost all the enzymes are proteins, but there are certain exceptional cases. And if we divide that enzymes depending upon the chemical composition, then that will be divided into the two main types. One are the simple enzymes and another are conjugated enzymes or the conjugated enzymes. Now, simple enzymes are just made up of the proteins. They are not having any extra groups so that it can contribute the reaction mechanism. But in case of a conjugated enzymes, it consists of the number of different types of groups. So these are called as the conjugated enzymes. Now here, so this is the conjugated enzyme because this is the main structure of an enzyme, which is the protein, and it also consists of one of the region or one of the group which is also important to catalyze the reaction. So the group which is attached to the enzyme so that it can also contribute to increase the rate of reaction. So the main part of an enzyme is called as apoenzyme, which is inactive, and unless and until it will bind to another group, it will not catalyze its reaction. So this is apoenzyme is inactive one, but it will, when it will be attached with the certain group, that may be any metal or that may be any organic group, then it will form the active enzyme. That active enzyme is called as a holoenzyme, and inactive enzyme is a apoenzyme, and these are the small groups which will require for the reaction catalysis. And so such a type of enzymes are called as a conjugated enzymes. So here it is the composition of an enzyme molecule. Uh, if that enzyme is a conjugated one, then it consists of the different groups, and that group is called as a cofactor. That cofactor may consist of the prosthetic group or coenzymes. And as we have discussed that holoenzyme is nothing but the inactive enzyme, that is apoenzyme, which is bound with the cofactor. And that cofactor may be any organic molecule or that may be any metal ion. Now sometimes enzyme will not properly work without some extra components, and such enzymes we have discussed that are called as a conjugated enzymes. And that extra enzyme, extra factor or the extra group, we have already discussed it as a prosthetic group or cofactor. Now that this is the apoenzyme, and that apoenzyme is attached with this certain groups. Here the metallic factor is there, here the coenzyme is a present, here the coenzyme and the metallic factor is a present. Sometimes it will require both coenzyme and the cofactor. So now it is the holoenzyme. If we remove this coenzyme and the metallic factor, then it will be only a coenzyme. Now, each enzyme has a specific site which is at which the substrate will bind, and that site is called as a active site. So, there are different types of simple enzymes. For example, pepsin, trypsin, urease. These are the simple enzymes which doesn't consist of any extra group. But if you consider the conjugated group, you will observe there are number of enzymes are there. For example, phosphoprotein, for example, a casein which is present in the milk. Then glycoprotein, for example, the insulin which is a glycoprotein. Then nucleoprotein, it is present in the with the DNA, that is a histone. Then chromoprotein is there. Then lipoprotein is there, which consists of the lipid moiety or lipid group. Flavoprotein, which is attached with the flavin group. Metalloprotein, the proteins which are attached or it consists of the metallic group as a cofactor. So such a type of enzymes are called as a coenzymes. Now we have discussed in little bit about the cofactors which consist of the prostatic group, coenzymes and the metal ions. Now what about the prostatic group? This prostatic group is not always the protein, but it is tightly bound to the enzyme, and when it will bind to the enzyme, it will form the holoenzyme, and it cannot be dissociated properly from the enzyme. For example, if we consider the hemoglobin, hemoglobin is not an enzyme, but hemoglobin is a protein, and in hemoglobin there is a globular protein which is attached with the heme as a prostatic group, and so this is the mechanism, or this is the example which indicate that how the prostatic group will attach to the enzyme. Now, what about the coenzymes? So, the coenzymes are the non-proteinic molecule, but these are the organic, that is a carbon-containing molecule, and these are the small in size than the enzymes, and these are loosely bound to the enzyme. Actually, prostatic group is bound very tightly to the enzyme, but these coenzymes are bound very loosely to the enzyme. So, one can easily set any reaction, or during sometimes it can easily remove from the enzyme. So it is just a temporary state of an enzyme whenever the coenzyme will bind to the enzyme. So there are certain examples of a coenzyme. Generally these are derived from the vitamins. Here there are certain examples, that is the riboflavin, thiamine, folic acid and number of water soluble vitamins which will act as a coenzyme for number of enzymes which are present in our body. Now there are different types of metal ions which are required for the different enzymes in order to catalyze the reaction. So these are the inorganic molecules or the inorganic cofactors, and usually they function as activator, or they will increase the rate of reaction, and sometimes they also act as an inhibitor, that is sometimes they also decrease the rate of reaction. So number of cofactors, as I discussed that number of cofactors will required for the different types of enzymes. For example, carbonic anhydrase enzyme will require zinc as a cofactor. Alcohol dehydrogenase also requires zinc as a cofactor. Then cytochrome oxidase, which is present at the electron transport chain, which will require the copper ion as a cofactor. And pyruvate phosphokinase enzyme will require K plus and Mg plus plus as a cofactor. So how the enzyme will work? So it is a very simple, as this is the larger molecule of an enzyme, and it consists of the very specific site which consists of the active site, and the active site is the site of on the enzyme where the only substrate molecule will bound. And when the substrate molecule is a very specific to this active site, therefore the specific enzyme will act on the specific substrate. So when this substrate is acted upon by this enzyme by attaching at the active site, it will form the enzyme and the substrate complex. Now this is the transition state. It means that it can be reversed into this for enzyme and substrate separation, or it can also converted into the product form. And when the product is a form, the product will get removed from the active site, and now this enzyme will remain free to work or to act on the another substrate. Now this is the graphical presentation where the reaction will take place without enzyme and with enzyme. This blue color graph is indicating when the enzyme is active, it will decrease the activation energy. Activation energy is the energy which is required for the reactant to reach the transition state. So this is the just animated example. So here the reactants are present, and when I have to transfer this stone at this side, then I have to pick up this stone, I have to reach here, and from there I have to pull that stone. So this is the main chance, or this is the main point where there is a equal possibility that it can come here or it can come down their side only. And so this is the very important step as in this stage. So here also this stage is very important for the reaction catalysis. So this is the transition state. And so in order to reach the transition state from which the enzyme and substrate complex will can broken down into the enzyme and substrate, or the substrate can be converted into the product. And suppose this is the red line, or the red line is indicating that any reaction is taking place without enzyme, then it will require a lot of energy, activation energy. And then once it will reach the transition state, then it will convert the substrate into the product. But it will require the more activation energy. But if such similar reaction is catalyzed with this catalyst or the biocatalyst or the enzyme, then that enzyme will decrease this activation rate without taking part in the chemical reaction, and then it will reach to the transition state and it will convert that substrate into the product. So this is the just graphical example. Here it will indicate that enzyme will act during the enzymatic reaction by decreasing the activation energy as compared to the reaction which require the enzyme, which is without enzyme. The reaction which is catalyzed by the enzyme will require the less activation energy. Now, how the enzyme lowers this activation energy? There are certain points which has to be considered here. One is whenever the reactants are present along with the enzymes, then enzyme will take reactant out of the solution so that the activation energy will get decreases. Then another mechanism is the enzyme will establish the weak bond between the reactant and the enzyme. Again, it can also decrease the activation energy by bringing closer together that reactant molecules. Again, it can also form the bond strengths in the reactant, and also it can form the transition state. And by considering these 5 steps, it is clear that how the enzyme will decrease the activation energy. So, in this lecture, we have discussed about the enzymes. We have studied about how they catalyze the reaction with the suitable example, and we have also discussed about the simple enzymes and the conjugated enzymes. We have discussed about the prosthetic group, what are the cofactors, what are the coenzymes, what are apoenzymes, what is holoenzyme, and how the reaction is catalyzed by decreasing the activation energy of the reaction. So I hope that you have understand about this concept of enzymes. Thank you so much.